The schooling of T cells in the thymus involves a strict selection process in which only 1 to 3% of thymocytes succeed in survival and export. Thymocytes dynamically migrate across different regions of a thymic lobule, each representing a unique stromal environment, in order to experience key events in development. The most notable of these regions are the corticomedullary junction, the cortex, the subcapsular zone, and, finally, the medulla. The following animation depicts the intercellular communication that regulates thymocyte trafficking and development. In the postnatal thymus, circulating progenitor cells migrate into the thymic parenchyma through the enriched vasculature that surrounds the corticomedullary junction. Their entry is regulated by adhesive interactions between platelet-selectin glycoprotein ligand 1, PSGL1, expressed by the progenitor cells, and P-selectin, expressed by thymic epithelium. Once in the parenchyma, these cells begin their initial development through the double negative or DN stages. DN thymocytes require notch signaling for their initial instructional cues, as well as for guidance throughout the cortex. They migrate to the outer region of the cortex, regulated by chemokine signals through CXCR4 and CCR7. Further outward migration is mediated by CCR9 signals. Through this movement, DN thymocytes begin to assemble the cell surface pre-TCR complex. Successful formation of the pre-TCR complex initiates cellular proliferation, signals for further development into double positive or DP thymocytes that express both CD8 and CD4 coreceptors, and finally, signals for completing the assembly of the TCR complex. DP thymocytes generated in the outer cortex are motile, interacting through their TCR with peptide MHC complex expressed by cortical thymic epithelial cells. This interaction begins the process of negative and positive selection. In the first form of negative selection, high avidity interactions elicit signals that lead to the deletion of thymocytes through apoptosis. Additionally, most cortical DP thymocytes fail to receive TCR signals and are also destined to die at this stage. Positive selection occurs when DP thymocytes are induced to receive signals for survival through low avidity interactions. Only 3 to 5% of thymocytes survive this checkpoint of T-cell development. Successful positively selected DP thymocytes gain expression of CCR7 and differentiate into CD4 and CD8 single positive or SP thymocytes, through which cells are attracted to the medulla. SP thymocytes that have migrated to the medulla spend approximately 12 days there before being exported from the thymus. During this period, central tolerance takes place, and SP thymocytes are further selected by dendritic cells that promiscuously present self-antigens. These self-antigens are supplied by the ever-important air-expressing medullary thymic epithelial cells. As modeled here, this selection process leads to the deletion of tissue-specific antigen-reactive T cells and the generation of regulatory T cells. Finally, at the end of this highly selective and arduous journey, mature thymocytes are attracted back to the corticomedullary junction, where they travel through the perivascular space and are channeled to postcapillary venules. These T cells are then released into the periphery, equipped with the necessary knowledge and tools to mount an immune response.